in drug discovery, diagnostics, and process technologies. With 13 drugs and over 80 technologies licensed to industry, CDRI conducts cutting-edge research in various disciplines. And to guide this prestigious session, now I have the distinct honor of introducing our esteemed session chairs, Dr. Kamaljeet Chopra and Dr. S.H. Sharma. Foremost, I would like to introduce Professor Kamaljeet Chopra, who is a prolific researcher from University Institute of Pharmaceutical Sciences, particularly in the field of neuropharmacology and metabolic disorders. Known for a strong citation record, she is known for her impactful contributions to experimental pharmacology. We have Dr. Sharma with us. He is HOD Department of Pharmacology and Toxicology at Naipur Mohali. Sir did his PhD from AIIMS New Delhi. He has been bestowed upon various awards including OPPI Award and Amir Chand Award. He is a fellow of International Pharmacological Society, and he has published more than 150 research and review publication in the peer-reviewed journals. May I request our session chairs to please come on the dais, guide the session by formally presiding and introducing our orator, Dr. Kavita Gulati. Please welcome Dr. Kavita Gulati with a warm round of applause.
Hello. Good afternoon, everyone. I think that's nice to see actually after post-lunch session. I can see many of you. That's great. You have come for listening to Professor Kavita Gulati. She's Director Professor of Pharmacology at Vallabhbhai Patel Chess Institute, University of Delhi. With over 3 decades of teaching and research experience, she has significantly contributed to clinical and experimental pharmacology, toxicology, and cardiorespiratory pharmacology. She has received the Achari Prize, Omnas Prize, and Professor B.N. Ghosh Oration of IPS, and she coordinated ADR and Medical Device Monitoring Center at VPCI and served as Nodal Officer of DHR Multidisciplinary Research Unit. She has over 200 publications, and you have seen during the inaugural session, one of her book was launched actually. So that's also congratulations to you.
Thank you so much.
With these words, we'd like to invite you to give a oration talk.
Thank you so much, Dr. Sharma, for the kind introduction. So first of all, I thank the jury, the president, and the executive committee, and the organizers of this conference to give me the opportunity to deliver this CDI oration at this forum. So I will be speaking on synergizing modern medicine with traditional wisdom in new drug discovery. So what is the traditional system? The traditional system is the world's oldest system of healthcare, and it is the sum total of knowledge and experiences gained over the centuries of years through the practices and the theories of some traditional system in their cultures. So this is the knowledge which was propagated through word of mouth, and it proved itself and continues. So in many countries, it is known by the different names: complementary, alternative, non-conventional medicine, and it differs philosophically from the modern medicine, as it depends mainly on the holistic approach, that is the balancing the mind, body and the spirit, and it has the nature-based, holistic and personalized approach, because it varies depending on the basic constituents, characteristics, gunas of the person. So it has the personalized approach, which the modern medicine has also started based on the genetic studies. So by WHO, till date, more than 75% of the population depends on traditional medicine. On the other hand, what is modern medicine? That is the contemporary, allopathic, conventional, which relies on evidence-based medicine, and it has the proper standardized dosages, treatment schedule, and everything is fixed up. So it is usually symptom-based, single-targeted, and now we have following the multi-targeted approaches as well. So it is a standardized form of healthcare when drug dosages, formulations, and everything is pre-designed and is standardized depending on the evidences. So what is the need of integration of the two systems? Because in modern medicine, we screen millions of molecules through combinatorial chemistry, and then finally, after going through all the stages and by the laborious work, we come to the attrition of some of the drugs. So, but in traditional medicine, we have the molecules which are nature-based and well accepted and have been shown to be empirically efficacious and safe on thousands of years. So we can have those molecules to go forward for the drug delivery. Then traditional formulations are usually polyherbal with the multi-targeted approaches. So they could be good alternative or adjuncts in the complex diseases like diabetes, cancer, neurodegenerative diseases. So they can provide adjunct to the mostly single targeted drugs in the modern medicine, but the tradition often lacks the standardization. So this can be achieved by modern analytical and pharmacological methods which can refine and validate these formulations, ensuring evidence-based efficacy and safety. So integration could revolutionize drug treatment in several otherwise intractable chronic pathophysiological states, and this can give the efficacious, safe, and evidence medicines and synergize each other. So we have clear-cut rationale for integration of the two systems, one depending on the personalized medicine, holistic approach, nature-based, and one which focuses on disease diagnosis, evidence-based therapies, and technological advancements for acute and chronic diseases. So when we integrate these two, we can have the modern medicine which can provide leads for innovative drug discovery and integrative healthcare. So what are the approaches to integration? Basically, these are of two kinds. One is from bedside to bench, and one is from bench to bedside. So the top three, that is polyherbal formulation, standardization, reverse pharmacology, and ethnopharmacology, follows the bedside to bench approach, and the system biology-based approaches using the omics, isolating the active principles, applying all the omics, genomics, transcriptomics, finding the targets and developing the drugs, or using the chemical informatics and AI-assisted screening. So basically, in the reverse pharmacology approach we usually follow in this integration, which was followed by Professor Ramnath Chopra, who is also known as the father of Indian pharmacology, who was among the first scientists to do the study on the crude extract of raw Wolfia serpentina in 1930s from this Ayurvedic plant. He demonstrated that this plant has tranquilizing effect, sedative effect, plus the hypotensive effect. And at that time, no good drugs were available for hypertension. So this inspired the Western world, and they did, the Ciba-Geigy, the Swiss scientists, did the studies. They extracted the reserpine and developed the drug Sarpasil from our Sarpagandha plant. So this Indian medicinal plant was given global recognition, and this shows how combined efforts bridge the gap between the traditional knowledge, wisdom, and the modern pharmacology. And usually in this traditional system medicine, we have the polyherbal formulations. They believe on that the one component may counter or complement the side effects of the other component, or they can complement the other plants. So usually they have the polyherbal constituents which are generally multi-targeted. So in that case, one has to do the quality, safety, and efficacy needs to be ensured, and the steps involve identification of raw material, because if the raw material changes, it depends on the seasons, what soil it has been taken, and different conditions. So we have to standardize the raw material, then do the phytochemical profiling using the latest technologies, quality control parameters depending on the physical parameters, microbial load, biological standardization. So example is the standardization of Chyawanprash and the Shmularisht will increase the efficacy and acceptability of these two formulations. So last from 25 years, we are working on the integration of various traditional medicine with the modern medicine. I will be discussing, giving the just view of some of the studies we have done with different traditional systems. So I'll be discussing first the Unani medicine. Unani system has a long history and it is holistic based on the 4 humor theory of Hippocrates and has a rational अभी अभी तो चल रहा था अभी शायद आ गया हाँ नाउ इट्स ओके हाँ So as per Unani medicine, if there is imbalance between the black bile, yellow bile, blood and phlegm, it results in the sickness. So the maintenance of this balance is necessary. So I'll be discussing with reference to the respiratory diseases, as these are amongst the most common cause of morbidity and mortality. And asthma is one of the very prevalent condition. So current therapeutic strategies involve corticosteroids or the bronchodilators. But they are accompanied with some of the side effects, or we have the refractory cases to the steroids. So always we are in search for the alternative approaches. Traditional medicine system could provide adjunct therapeutic modes for effective management. So we did a clinical study to validate the efficacy and safety of Unim 352, which is a coded compound, and is given in the Unani system of medicine in their clinics in patients of bronchial asthma. So we did a collaborative study with CCRUM, Central Council for Research in Unani Medicine, on a polyherbal preparation. It was long back, we did this study in 2012. And CRIUM prepared a standardized, authenticated preparation of this compound. And they also prepared a placebo for giving to the patient so that it can be the single blind study we did. It has these 6 constituents. So we followed all the guidelines of the GCP, Institution Animal Ethical Clearance was obtained. And after discussing all the details of the procedures and the requirement from the participants, we got the informed consent form. And the subjects were the bronchial asthma patients from OPD of Patel Chest Institute. And they were diagnosed by pulmonologist, and they were enrolled as per the inclusion-exclusion criteria. Informed consent was taken. The mild to moderate persistent asthma patients were taken, and no comorbid conditions, no medications. So after randomizing, as per the computer-generated randomization chart, they were grouped in 2 groups. One was given the conventional treatment with the placebo, and the other group was given the conventional treatment plus Unim 352 Majund, 10 grams twice a day. In both the groups, SOS emergency levosalbutamol is given, and they were followed up for a month, 3 months, and after every 4 weeks, they were followed up. The study markers were the forced expiratory volume 1 second, which is the most important parameter in a bronchial asthma patient, the FVC ratio, incidence of asthma aggravation, frequency of emergency bronchodilator use, clinical symptoms, and these parameters were the outcomes. So we see that basal FEV1 significantly increased in the patient who was given Uni 352 along with the conventional treatment. At 12 weeks, there was improvement, significant improvement in both the groups. However, the magnitude of the response was more in the group which was given adjunct therapy with UNIM 352, thus suggesting that it enhances the kinetics because the effect which we observed in alone conventional group at a significant level at 12 weeks was observed in 6 weeks, and moreover the quantum of the response was also increased. Similarly, the change in FEV1 was also significantly improved in this group. It is the simplified graphical representation of the same which is being shown there. And this was supported by the fact that the percentage of the patients who were giving the incidence of exacerbation, they were also significantly reduced more in the group which was given UNIM 352 along with the standard conventional treatment. And it was also supplemented by the finding of the absolute number of SOS bronchodilator use. So after validating the compound by using the modern methodology, we showed that yes, in the Unani system of medicine, what is being practiced is really observed in the modern medicine also, as per the ethical committee, because we cannot give it as alone. So it has to be given in the conventional treatment, but we came to know that it improves the kinetics as well as the quantum of the response to conventional drug. Secondly, it also reduces the need of the SOS bronchodilator, so it can be economically also good for the patient to if we give it as an adjunct therapy. So after this study, we got another project from the CCRUM to do the preclinical studies to evaluate the cellular and molecular mechanisms involved in anti-asthmatic effect of UNIM 352. For that, again, the modern methodology, which is the well-established and widely accepted, all those markers were used to establish the efficacy of this drug and to evaluate the mechanism of action. And it was evaluated all the aspects of bronchial asthma, because we say that bronchial asthma is a chronic inflammatory disorder with bronchial air hyperresponsiveness and narrowing of the airways. So at all the aspects, we studied the effect of UNIM 352, that we established two different models. In one, the airway inflammation was studied, and in this oval albumin along with aluminum hydroxide was given. After the sensitization on 14 days, the challenge was given, and the changes in the airflow and was recorded, and the blood and the bronchial alveolar lavage fluid was collected to see the various cytokines which are the markers of the immunological response, inflammatory response, and as such also in the whole body plethysmograph in which the animals is kept, and the difference in the volume of the chest with the compartment where the animal is kept, that gives the directly value like the spirometry, that is the peak expiratory flow rate, and it gives the airway hyperresponsiveness and airway constriction in the form of the P-enhanced ratio. P-enhanced was measured, and it also showed P-enhanced is measured in response to the spasmogen, like methacholine is given to the animals at different doses, and the P-enhanced value is taken, that how much of hyperreactivity of the airways is there. So it was also found to be reduced by the UNIM 352, and the results were comparable with the standard drug. And for the other model was for the airway remodeling, in which the higher doses of oval albumin was given with a high dose of aluminum hydroxide for a longer duration, and the daily aerosol 2% of the oval albumin was given challenge for 22 days, and the animals were administered with the standard drug. So we found that as compared to the normal healthy animals, the experimental control, that is given the oval albumin sensitized and challenged rat, there was significant increase in the inflammatory marker, pro-inflammatory marker TNF alpha, which was reduced significantly by both the doses of UNIM 352, and was also the results were comparable with the prednisolone. And similarly, these effects were replicated at the systemic level also. So we see that what is happening at the local level, that is at the bronchial alveolar lavage fluid, what we recorded, that was also seen at the systematic level. So similarly, IL-4, which is a cytokine released from the Th2 cells, was also found to be increased significantly as compared to the normal healthy animals, which was also taken care by the pre-treatment with the UNIM 352. And similarly, although at very low levels, but the similar kind of responses were observed in the blood. So this shows that this drug is really effective, and at both the compartment, this effect was seen. And because we know that IL-4, when secreted from the Th2 cells, it increases the IgA production and causes increased infiltration of the airways by the elementary inflammatory cells, this is eosinophils. So we see that there was increase in the neutrophil and the eosinophil cell counts, which was reduced by the higher doses of UNIM-352, and the results were comparable with the prednisolone, again standardizing the model of our model of airway inflammation. And similarly, over specific IgE was also found to be increased, which was reduced in the animals which were given UNIM-352 for 14 days. So all these effects together showed that UNIM-352 has the cellular effects by reducing the inflammatory cells infiltration in the airways, as well as at molecular level it is affecting the cytokines by affecting the immunological responses and inflammatory responses. So in the model of airway remodeling, we studied the markers as defined by the modern system medicine. In pharmacology we study, and the TGF-beta was found, which is a profibrotic factor and increases the collagen deposition, was increased and was reduced by the higher doses of UNIM-352 in bile as well as the blood. And similarly, hydroxyproline was measured in the lung homogenates, and it was also found to be increased and taken care by the UNIM-352 administration. These levels were also supported, these findings were also supported by the levels of histone deacetylase 2, which is known to suppress the expression of inflammatory gene. So these levels were upregulated in the animals which was given the UNIM-352. So usually in pharmacology, you must have seen we conduct a battery of tests to confirm a finding. So because on one parameter if we are getting and another parameter we are not getting, it may be by chance or some other factors are involved, or some particular one mechanisms may be involved. But all these factors, when you take a battery of tests, they complement each other and support the findings and strengthen the results of your study. As when we see the oxidative stress marker, which is, is one of the very important precipitating factor for bronchial asthma, was measured, and we see that MDA, which is the product of the lipid peroxidation by the superoxide, peroxide, all the free radicals, was increased in disease control animals, and which was reduced by the higher doses of UNIM-352 and was comparable with the standard antioxidant ascorbic acid. And similarly, SOD and GSH, which are the antioxidant superoxide dismutase, antioxidant enzyme, and the reduced glutathione was also altered, suggesting that in this, our model of bronchial asthma, that is airway remodeling, there was oxidative stress, which was reduced by this Unani polyherbal formulation. So all these results were substantiated by the histopathological studies of the lung tissue, which clearly showed that there was increase of infiltration by the eosinophils, neutrophil cells, goblet cell hyperplasia, and subepithelial fibrosis, which was found to be less and in UNIM-352 and the prednisolone. So they substantiate our earlier findings, which we observed on the markers of inflammation and immunity and the tissue remodeling. So this suggested that UNIM-352 acted as an anti-inflammatory, immunomodulatory, anti-remodeling, and antioxidant agent. So the preclinical studies elucidated the underlying mechanisms, thereby supporting the clinical findings of the traditional medicine in bronchial asthma patients. So for the first time, this study was presented in 6th European Congress of Pharmacology in Spain, and in very prestigious 17th World Congress of Basic and Clinical Pharmacology, Cape Town in South Africa, and others are the publication from this research work, these two research works. Then I come to another example, evidence-based example of another medicine from the Ayurvedic medicine, which is again more than 3,000 years old, and it focuses on holistic health and personalized medicine based on three dosha prakriti of the individual. Rich pharmacopoeia is there with thousands of herbal formulations, and even the CDRI has taken forward two of them, Bacopa Brahmi for the memory enhancer, Mukul for the gugulipid for lowering the lipid levels. So strategic integration with modern science can facilitate safe, evidence-based drug development. So we did a preclinical study to evaluate Vidania somnifera as adaptogen during stress and the probable mechanisms. Stress, as defined by Hansele for the first time in 1933, is any external or internal stimulus that disrupts physiological homeostasis, and the ability to adapt is critical and for survival. So stress responses involve complex neurohormonal chemical pathways in the CNS, and the stress is a highly interactive phenomena. Multi-system involvement is there. Almost all the body systems are affected by the stress, whether it is CNS, neuroendocrine, immune, cardiovascular, GIT. So there is a therapeutic gap in the management of stress-related disorders, and multi-targeted therapies are being sought which are effective and safe. On the other hand, in Ayurveda, there is a mention of Vidania somnifera, which is recognized as a rasayana or adaptogen, which means a natural agent that enhances resilience to physical and emotional stress and supports overall vitality. It attenuates the negative physiological and psychological effects. So experimental studies were conducted to evaluate the effect of Vidania somnifera and its probable mechanisms. So we studied the neurobehavioral responses because stress induces anxiety, and which was done by the EPM, again widely accepted, widely used and well established model to measure the anxiety in the EPM, and we find that the percentage open arm entries were significantly increased in the patients who, in the rats who were given the restraint stress. Again, restraint stress is a model to produce the emotional stress in the animal. And Vidania somnifera was not producing any significant effect in rats which were normal, suggesting that it believes in maintaining the homeostasis balance, and when the levels are affected, it increase the percentage open entries. Similarly, the neuroendocrine parameter, the corticosterone, which is found to be increased in stress, was found to be reduced by the Vidania somnifera at the higher doses. We also measured the adaptive immunity by measuring the cell-mediated as well as the humoral immune responses, which we know are reduced in restrained stress animals, and the Vidania somnifera in higher doses produce a positive effect and elevated the immune responses, because in stress, when the immune suppression is there, there is the increased susceptibility to infections, and the CMI is also reduced. So we are also working a lot on the nitric oxide, which is a signaling molecule and present in several brain regions which are known to regulate the stress responses. Further, NO also acts as a neuromodulator, interacts with various neurotransmitters, and exerts multi-system physiological effect. So there was a similarity between the stress effect. It was also involving many neurotransmitters, all the systems in the body, and nitric oxide was also working on almost all the body systems. So we thought that whether alteration in NO signaling during the adaptogenic activity of Vidania somnifera was investigated to evaluate the role of nitric oxide during the adaptogenic effect. So we see that Vidania somnifera, as such, in recent stress, there was reduction in the, because nitric oxide has a very low half-life, so we measured the stable metabolites of nitric oxide, nitrates and nitrates were found to be reduced, and we have many papers on this. And it was accompanied by increase in MDA and reduction in the GSH. And Vidania somnifera affected this, and it reverted these levels towards the normalcy. And to see the involvement of nitric oxide, we measure the pharmacological tools, which is a standard technique in pharmacology. We gave the precursor of nitric oxide, that is L-arginine, which is oxidized to nitric oxide plus citrulline. And the dose, which was sub-effective, with Vidania somnifera 100 milligram per kg, effect was seen to be becoming efficient in producing the increasing the levels of nitric oxide and decreasing the oxidative stress. And LNM, which is one of the nitric oxide synthase inhibitor, was able to reduce the effects of the 400 milligram per kg of the Vidania somnifera. This shows the effect on the immunological markers, IL-4 and interferon gamma, again from the
Th2 cells and from the Th1 activity T cells and the natural killer cell which were reduced were found to be increased by this, and similar kind of interaction was observed with the nitric oxide precursor as the nitric oxide synthase inhibitor. So we summarize that RS induced neurobehavioral, neuroendocrinal, immunological responses, and these were attenuated by Withania somnifera. Further, there was a restoration of the oxidant-antioxidant balance. L-arginine potentiate effects of Withania somnifera on immunological and biochemical parameters, and NOS inhibitor suppress these effects. So it was inferred that Withania somnifera has adaptogeny effect, and nitric oxide may be involved in mediating such effect. And now we are doing studies to, because in nitric oxide synthase inhibitor was the general, INOS, ENOS, NOS has the different effects. And nitric oxide, which at the low doses act as an antioxidant, when its levels are increased, it becomes a pro-oxidant. So there is a critical balance between the two, which affects the final homeostasis of the body. So we are working on the gene expression of INOS, ENOS, and NOS, that what systems are involved in the mediating the effects of the Withania somnifera. So these are the publications from the Withania somnifera studies. And actually, we have done later on the studies with Withania somnifera in type 2 diabetes, mellitus-induced cognitive impairment, in overwhelming-induced bronchial asthma, in post-traumatic stress disorder. So all this is the published work with this Ayurvedic medicine. Then I come to the third system, that is the yoga, which is upcoming field, and it is based on concept of uniting the mind, body, and spirit to achieve a level of maximum energy and efficacy, and benefits are being acknowledged globally, as evident from the celebration of the International Yoga Day on 21st June every day since 2015. So it is a contemporary area of extensive biomedical research to generate scientific evidence and develop skills to bring it in mainstream in healthcare system. Again, I'll be discussing with reference to the respiratory system, and it has yoga because it has the beneficial effect at both the levels, at the respiratory as well as the calming effect on the brain. How could it affect the management of the bronchial asthma? Similar studies, as I mentioned, this is the later, this we did in 2017 in bronchial asthma patients, and in 2020 in COPD patients, and we find similar kind of very encouraging results.
Dr. Kavita, can we have the conclusion?
Yes,
of
just
this?
I will finish in the 3-4 minutes.
Okay.
Yeah. So similarly, it was randomization was done. All the modern pharmacology techniques were used, guidelines were used. The yoga schedule was prepared in consultation with the director of the Morarji Desai National Institute of Yoga. And the patients were taught by a trainer for 2 weeks the yogic intervention. Then they were asked to maintain a diary, and the follow-up was done. And all the parameters, like latest parameters, like FeNO we included, which is a novel marker in which the patient has to breathe in a sensor. And directly we measure the levels of nitric oxide, fractional exhaled nitric oxide was measured, and the effect of the yogic intervention was seen. And again, we see that the magnitude of the response was much better in the group which was given the adjunct therapy with the yogic. And similar kind of results were observed in the immunological and oxidative parameters. Asthma quality of life questionnaire is there by the Elizabeth Juniper. It was used, and it has mainly the 4 domains: symptoms, activity limitation, emotional function, that is a psychosocial response of the patient to conditions, and environmental stimuli. So this was the questionnaire. So we find that the group which was given adjunct therapy, yogic intervention, showed the better effect, although the conventional treatment alone also produced the significant effect in 3 months, but all the 4 parameters, including the emotional function as well as the environmental stimuli, that is the resilience to environmental stimuli, allergy to different pollens, or was also improved in the patients who were given yogic intervention daily for 50 minutes for 3 months. So we see that the introducing yoga as adjunct therapy ameliorated the kinetics and quantum of response to the standard medications, reduce need of SOS-SABA, so can be economically beneficial for the patient as well. So such translation research could help in the integration of traditional wisdom and modern medicinal concept for the sustainable management of chronic disorders. These are publications from this yogic intervention and these clinical studies on COPD and the bronchial asthma, the 2 studies we did. Actually, then we did one more study, but I won't be going in details of this. But like these were the efficacy studies. We did drug-induced toxicity studies also, in which ATT, produces the hepatotoxicity. And in this, the vowel curcuma was given, and we found that it was effective in reducing the macroscopic, microscopic, histopathological, and all the effects of the ATT drug treatment. And then we did the toxicity studies, and now actually CCRM has invited us to give a protocol for doing a clinical study in, with CCRM against ATT-induced hepatic dysfunction. So we are preparing a protocol for that, for doing these clinical studies. So these are publications from this drug-induced toxicity studies, and we did in paracetamol against the alcohol and all. So finally, I'll conclude that all these evidence collectively illustrate that integrating traditional knowledge with modern scientific validation can provide evidence-based support for time-tested therapies. So such integration can enhance the efficacy and safety of current pharmacotherapy, reduce treatment-related adverse effects, and enable the development of affordable, multi-targeted therapeutic options for chronic diseases. So meaningful synergy between modern pharmacology and traditional wisdom represents a forward-looking strategy in new drug discovery, one that is scientifically robust, clinically relevant, and globally significant. So I acknowledge the Ministry of Ayush for funding the work on the vowel curcum, on UNIM 352 clinical trial, as well as the preclinical studies, CRIUM for providing us with a placebo and the standardized authenticated preparation, ICMR for funding the Vidania somnifera work, Dr. Amit Agarwal providing the Vidania somnifera extract, standardizing it. Professor Ere was
Thank you,
inherent
Dr.
part
Gulati.
of all the
I
studies.
think we will have to
Okay,
stop
thank
now.
you so
Yeah.
much. And all these students
And-
who have done PhD and MD in these topics.
And
Thank you so much.
Thank you for very nicely demonstrating that how the synergy of Unani, Ayurvedic, and yoga, along with the using the scientifically validated models that you have used, using
Yeah.
a battery of molecular markers, how that has a potential
Yes.
to really result in.
Yes, thank you so much. And like, actually, I want to emphasize that when we are having cough for more than 1 month, we say to each other, take honey with pipernigram. In our hearts, we have some belief of others. So it is not time to prove, it is now time to implement
Very
the
right.
synergy
Using
of
all
the
the
two.
reverse
Yes.
pharmacology approaches.
Yes, yeah, at least whatever approach you use, system biology approach, the AI-assisted STEMCAL approach, but we need to now, it's high time to synergize the two. Thank you so much.
Congratulations, Dr.
Thank
Gulati.
you.
I think 30 minutes are not enough to present 3-decade work actually. So that's why we have to stop. Thank you very much.
I request our session chairs to now felicitate ma'am with a token of appreciation. Please, ma'am. I'll come. We would now like to felicitate our mentor, the motivation behind we will now felicitate our mentor, the motivation behind this mega event, Professor Kamaljeet, for seamlessly conducting the session along with Professor Sharma. And we will be holding back felicitations for Professor Sharma as he is committed to chair the next subsequent oration too. We are grateful to you, sir. Thank you. Our next oration, Dr. S.C. Lahari Oration, is a prestigious academic award established in honor of Dr. S.C. Lahari, a pioneering Indian pharmacologist. This oration is conferred on distinguished researchers who have made significant contributions to pharmacology and related biomedical sciences. By recognizing scientific excellence, it commemorates Dr. Lahari's legacy and promotes continued advancement in pharmacological research and education. For chairing this session, we have Professor Sharma already with us. I would like to invite Dr. Sukhbir Kaur to join him on the dais. Your presentation, okay. So, now I request our session chairs to introduce our orator.
Hello. Good afternoon, everyone. And thanks to the organizers for giving me this opportunity to chair this session. So the speaker for this oration is Professor Sadhana Sathai. She is a senior professor of pharmacy at the Institute of Chemical Technology, Mumbai. She holds a PhD in pharmacology, M.Pharma in clinical pharmacology from Grant Medical College 1986 and B Pharmacy in pharmaceutical sciences. Her research focuses on the development of herbal-based therapeutics for CNS disorders, pharmacokinetic pharmacodynamic modeling, metabolic diseases, immunomodulation, and phytochemical anti-cancer approaches. Professor Sathai has supervised over 100 postgraduate and doctoral students and collaborated with leading institutions such as King's College London and IIT Bombay. A fellow of Maharashtra Academy of Sciences, she is a recipient of DST Vidushi Fellowship and multiple Best Teacher Awards. With this brief introduction, I welcome you, ma'am, to please give your presentation.
Yeah. Thank you, everyone, and good afternoon. I would like to thank the organizers for inviting me to give this presentation. My title of my presentation is From Blood to Brain, a translational strategy for early diagnosis and targeted therapy in Parkinson's disease. So, I would like to dedicate this presentation to Professor S.C. Lahari, who is a renowned neuropharmacologist who worked in the area of venom, immunology, etc. So, about Parkinson's disease, if you know about it, that it's a really frustrating disease wherein you find many movement disorders which make your life difficult. So, current status of this neurodegenerative disorder wherein there are neuropathological features. You can see that there is an alpha-synuclein, which is a protein, otherwise good protein, which helps in synaptic transmission, that gets misfolded. Normal monomeric form gets misfolded into beta pleated sheets, and that starts precipitating out, and that produces neurodegeneration and neuronal loss. So because it gets precipitated out, the microglia and the astrocytes, they get activated and there is a blood-brain barrier leakage and neuroinflammation. And if one cell dies, when there is a one neuronal cell death, particularly in the area of SNPC, substantia nigra, many other cells also progressively degenerate, and thus the disease is progressive, complex and multifactorial, particularly affecting the body movements, simply because dopamine controls the different body movements. As far as clinical diagnosis is concerned, the diagnosis is mainly based on symptomatic, meaning symptoms which appear, and 70% neurodegeneration when it appears, the symptoms start appearing. So, it's very difficult to diagnose this particular disease at an early stage, diagnosed only when 70% neurodegeneration takes place, particularly by all these invasive technologies, SPECT, PET, DOPA-PET, CT, etc. So as of today, there is no reliable, easily applicable and cost-effective diagnosis, and of course there is no cure. So it's the need of the hour that we diagnose this disease at an early stage so that we can intervene, we can have disease-modifying agents for early intervention to manage this disease and give a good quality of life to patients. Now, what do the stalwarts, they say? Professor Ole Isaacson from Harvard Medical School, he says that blood-based alpha-synuclein signatures could revolutionize the diagnosis of Parkinson. And Professor William Langston from Stanford University and CEO of Parkinson's Institute, he says that it is the only unifying thread in Parkinson's, which this is a target that holds a promise of true disease modification. So if you identify it early, you can give disease modifying agents in this case. So what is the research overview? So we produce polyclonal antibodies in rabbits with a high titer after each successive immunization, a high titer of purified antibodies specific to beta cross sheets, meaning the misfolded alpha-synuclein were achieved, and the antibody was further tagged with fluorophore, which could be optimized later in a NC-FLISA assay. The antibody specificity to the misfolded protein only and not to the monomeric form, because monomeric form is good, that was validated, and then the detection of alpha-synuclein protein, misfolded protein, using NC-FLISA was done in vitro and in vivo. So this was the proof of concept, like we wanted to do that so that we could go ahead. Now I'm just going ahead with this slide because I'm showing you in this. So this was confirming the selectivity of polyclonal antibody to beta cross-sheet form. So this is the beta, the green ones are beta cross-sheet form and the red ones are monomeric, that is good form. So we validated that our antibody only bound to the beta cross-sheets and not to the alpha cross-sheets, alpha monomeric. So that means that we have validated that it only binds to the beta pleated sheets, and see the sensitivity, it's from 1 picogram to 1000 nanograms. So you can say that 1 microgram per ml also could be detected, and the least that we could detect is 1 picogram. And if you see the blood levels of misfolded alpha-synuclein in the Parkinson's patients, it is about 30 to 50 picograms per ml. So we are quite in a, we are good as far as our method is concerned. Now, okay, now this was direct. Now when there is a misfolded synuclein in the blood sample, we have to use this competitive FLISA method. So this competitive FLISA method is particularly between the interaction of the misfolded protein in the sample, biological sample, whatever it is, and the one which is coated on the nitrocellulose membrane. And so it means that the increase in the concentration of the blood of misfolded protein in the blood serum will definitely show decreased fluorescence in the coated antigen. So this is what it confirmed. This is the fluorescence versus biological sample concentration of alpha-synuclein, and we could detect it, meaning in this competitive method. This is a little different method than the first one that I showed. It shows again very good sensitivity between 1 picogram to not 100, also 1000 picogram per ml, and the concentration in the blood sample of a Parkinson's patient which has been reported is 30 to 50 picograms per ml. Now we had to confirm it using in vitro and in vivo method. So in vitro method was sushi cells, wherein we had added rotenone, a known pesticide. It's a well-known model to create Parkinson's in animals. So we saw if it could produce the same markers in the sushi cells, and we saw that control shows highest fluorescence, meaning less misfolded protein, and at 24 hours after rotenone addition, the fluorescence reduces. At 48 hours, it is not detected at all. So this proves that you can detect the misfolded protein concentration in the cell supernatants, and similarly in the rat serum. The rat's brain was injected with either rotenone or misfolded beta sheets of alpha-synuclein. It was directly administered or injected into the SNPC area, and at different times, 7 days, 14 days, till 6 weeks, 42 days, we kept on taking the blood samples just to understand whether we are able to detect the concentrations of misfolded beta pleated sheets. And you can see that the sham control, where nothing was administered, it shows good fluorescence, meaning no misfolding. Whereas at 7 days, both rotenone as well as alpha-synuclein, it showed fluorescence, meaning, and a little less fluorescence than the sham control, meaning the protein has started misfolding because you have added something into the rat's brain area, and then slowly, slowly over the time, there is absolutely no detection of fluorescence, meaning the increase in the beta folded sheets is there. So this also proves the concept that we concentration of the beta cross sheets increase in reduction of fluorescence intensity, and the proof is proved. The POC is done both in vivo and in vitro samples. Now, okay, this was through the blood samples. Now, can we do IHC with the polyclonal antibody that we have produced? So we took the SNPC areas of the administered alpha-synuclein rat's brain and rotenone induced and sham control. You can see that beta cross-sheet induced rat brains show higher fluorescence. Again, this is direct method. So you are just detecting the fluorescence. Here, higher fluorescence meaning higher misfolding, and you can see that beta cross-sheet, it gradually increases. Even rotenone gradually increases, and sham control does not show any fluorescence, meaning where you have not added any misfolded beta sheets, you don't see any fluorescence, meaning no misfolding. So these results are just put here. This is confirming that not only from biological samples by competitive NC-FLISA method, you can even use this antibody to detect the misfolded alpha-synuclein in the brain samples as such for immunohistochemistry. Now, okay, so we understood that you can diagnose the disease at an early stage. You can diagnose as low as 1 picogram per ml of misfolded protein in the rat's blood sample for that matter. Then let us see what early intervention that we can do. So currently available treatments, if you see, it's only dopamine producing agents. Either it is dopa decarboxylase inhibitors like carbidopa, or it is COMT inhibitors like entacapone, or it is MAO inhibitors like selagelin, or something like amantadine that directly increases the dopamine concentration, or it is dopamine receptor agonist. So whatever it is the increase in the dopamine levels, and the relief is only symptomatic. And you all know, maybe if you have seen Parkinson's patients, those who are on this syndopa or dopamine kind of treatment, they always will have dyskinesia. That's the major side effect because of oxidation of dopamine in the body. So what next? So all that we need for health and healing has been provided by God in nature. That's what even Madam Gulati told us. So this is the same thing, all the challenges only to find it. So we were working on this antioxidant luteolin. It's not lutein, it's luteolin. It's a little different. And you get it in many of the vegetables. We have, it's a flavonoid, and we have worked on it on the Eclipta alba for treatment of epilepsy. And in the market, I have seen it in the US market, not in Indian market. It is a nutritional supplement for cognitive health. So generally people take it for antioxidant effect of it. I was interested in its anti-inflammatory and neuroprotective property, and mechanism is suppression of NF-kappa B and TNF-alpha lowering, and it has, because of its antioxidant properties and modulation of microglial activation, it produces neuroprotection as well. If you see the pharmacokinetics, although half-life is 5 to 7 hours and appears in the bloodstream in 1 hour, we have found that it is extremely lipid soluble. It's not water soluble at all, and it has got a very low permeability and brain biodistribution particularly. Now this is how the pure luteolin looks like, and this is what the formulation that we have developed in ICT looks like. We made it with help of a pharmaceutical technologist, and we are calling it Neura Health because it has shown good effect in vitro PD model in sushi cells and in rat model of PD. Now let us check what are the changes that occur in the brain. So if you see the normal brain, this is BDNF, which protects the brain, which produces healthy neurons. This is tyrosine hydroxylase, which is a rate limiting enzyme to produce dopamine in the body. This is tripeptide glutathione, which is an antioxidant, and these are two antioxidant enzymes which provide antioxidant effect. And this is KEAP1, it's holding on to another factor that's called NRF. So the levels of KEAP1 are low, NRF is good, antioxidant effect is good. And when you have Parkinsonism, when the alpha-synuclein starts misfolding and you have Lewy bodies, then there is a neurodegeneration in the SNPC area. And because of that, the KEAP1 increases, the GSH-SOD catalase will go down, the glial fibrillary microglia get activated. Because of this, you will have high inflammation and neurodegeneration as seen by the lipid peroxidase, high lipid peroxidase activity. So when this happens, the mitochondria get damaged. They are no more healthy, producing healthy movements. They are now damaged, degenerating the dopaminergic neurons and producing uncontrolled movements. So what? So we can determine if the Neura Health, which we have produced, is it better than luteolin alone, API? And these were the certain markers that we used. So I'll show you directly the results. So these were the results of in vivo efficacy of luteolin. So here we were just looking at the API, which showed antioxidant properties, reduced reactive oxygen stress, reduced KEAP1 expression, meaning increased NRF1, NRF2 and GSH-SOD catalase. LPO was reduced, indicating that neurodegeneration was reduced, cellular membranes were stabilized, GFAP levels were reduced, and we saw higher TH activity, tyrosine hydroxylase activity, so neurogenesis, and we saw less misfolded alpha-synuclein. So this was done by using our antibody. So neuroprotective, anti-inflammatory generation of dopamine-producing neuron. So everything that would help in modifying the disease. So we identified that we should work on this particular luteolin and make it more available to body. So we developed it for better bioavailability and brain biodistribution, and solubilization was achieved with using selected excipients, of course with a pharmaceutical technologist, Sutix people, and this is how the formulation looks like. We have filed a patent on this, which is also published, and we are awaiting the results now, from the patent office. Now, we have to see whether whatever that we have made is really available in the body, whether it increases permeability. So if you see the API release versus the NeuraHealth release in SGF and SIF, you can see it is 3 times higher release. 3 times higher release of the formulation in vitro methods in SGF and SIF. And in vivo PK studies indicated the Cmax was increased 1.5 times, the brain distribution was increased 3.5 times, and the Tmax came down by 1/6, meaning 36 hours to, this is what we found in rat. And now we wanted to see if along with this, whether it correlates with improved grip strength or motor coordination or locomotor activity, because that is what is affected in the Parkinson's disease. So whatever results that you get in PK, higher brain distribution, whether it really translates into pharmacodynamic activity. So we chose a model of Parkinson wherein we added rotenone into the brain of SNPC area of the rats, and these were the different behavioral parameters using rotarod, muscle strength and coordination, open field model for locomotor, mitochondrial health, complex 1 activity, GSH, all these from brain homogenates for antioxidant, and LPO from brain for neuroinflammatory. And these were the IHC markers, so BDNF, GFAP and tyrosine hydroxylase. This is what we saw from IHC. So, you know, whether the increased motor coordination correlates with antioxidant activity and neuroprotective activity as we visualize, and does the IHC confirm it? It's all what we wanted to see. So these are the results. I've just put it. You can see that rotarod, you can see 15% improvement by NeuraHealth, 30% improvement in locomotor activity, 60% higher improvement in complex 1, so more energy, better mitochondria, 85% of glutathione, increase in glutathione, which is a natural antioxidant tripeptide in the body, increase 37% and 54% of SOD and catalase, and 35% lower value of lipid peroxidase, indicating less cellular damage. You can see that BDNF increased 20%, which helps in normal healthy growth of neurons, and dopamine producing neurons 36% increase, and you can see that inflammation was reduced by 10%. So I can now say that it exhibits 10 to 85% better neuroprotective or anti-PD activity than the plain luteolin, meaning whatever that excipients we had added for better solubilization and brain biodistribution has worked here as seen by in vivo rat model. So the good news stories in medicine are early detection and early intervention. So yes, if we could detect the disease at an early stage, say 15 years early, and you can, give a patient. I'm not saying that the patient has to suffer, I mean, good if he doesn't suffer, but he can take, if he finds a higher level of misfolded alpha cyclin, he can take this luteolin for that matter. And so what is that translational strategy is that we have developed a novel polyclonal antibody. It was polyclonal, it's not monoclonal. With a simple setup at ICT, we have produced it, we have purified it by simple methodologies. We have confirmed its specificity only to the misfolded alpha-synuclein, and in blood samples also we could detect it in rats serum. And so it's a non-invasive, few microliters of blood sample, serum sample that we require to analyze this antibody. And then optimizing luteolin formulation for better solubility, bioavailability and brain biodistribution. So all that it did is, of course, we had kept a group of rats when we induced rotenone into the animal brain. There was a group where we had also started giving them luteolin formulation. So it improved motor coordination, attenuated oxidative stress and neuroinflammation, I just showed you in the previous slide, and dopaminergic neurogenesis. So early diagnosis and early intervention resulted into early neuroprotection. So this is a similar slide. I just want to tell you that the range of PD patients is between 30 picograms and 50 picograms per ml, and we could detect it at 1 picogram to 1 microgram, so wider range of detection. And yes, luteolin, uh, Neura Health showed better results than API. Now looking at the market potential, it's a easy and scalable technology, very easy and scalable technology with significant higher solubility and brain biodistribution, confirmed anti-Parkinson's and activity in preclinical cellular and animal models. And you can see the global market, almost 1900 billion US dollars market, which is expected to grow at 7.7% till 2033. So there's a huge market potential for this, and I'm so proud that recently I received a Vidushi grant from DST for early detection. So whatever proof of concept that we have done in the animals and in cellular studies, we've got money to study it in the clinical setup. So we have already connected and collaborated with biggest municipal hospitals in Mumbai, and we are taking it ahead to at least understand if the diagnosed clinical patients of Parkinson's do show certain levels of misfolded alpha-synuclein as against the healthy people for that matter. And of course, if the luteolin that we have made, that would take a little more time, but I'm going to use the normal luteolin that you get it in the market as a nutritional supplement. If that is given, is there any difference? So that's what we are going to see in with this funds that we have got. So thank you so much, and I'm happy that I finished it in time. And this is my team. She's Paramita, who actually developed, tagged, and studied this polyclonal antibody, confirmed everything. So she is Paramita, who is now working in Iceland in Alvotech Biotech company. And Priyanka was an MPharm student who worked on luteolin formulation and its confirmation in vitro and in vivo models. She is now currently doing PhD in America, St. John's University. So thank you very much. I'm open to all questions.
Yeah. Great. Uh, I really appreciate Professor Sadhna Sai for your excellent talk, actually, and excellent oration talk. I think you have really gave the idea from like bench to bedside. So that's one of the
Actually,
things
in the
actually
morning
like
somebody said,
translational
yeah.
point actually you have taken into consideration, and I've seen that you have done in vitro studies, then in vivo, and then going into the human with history with luteolin formulation with Neura Health alone and their combination. I think excellent work. I think we must give a
Thank
big
you.
hand.
Thank you so much. So, in the morning there was a speaker. She was a clinical pharmacologist, and she said, what's the use if you, like study so many things and it just remains in your thesis? So you have to see to it that it sees the light and we are able to help patients in some way or the other. So that was the motto. Thank you. Any questions? I'm open. Yeah.
Yeah, sorry. Actually, I think in oration session,
I'm
you can
That's my habit
you
of
can interact with actually the speaker after this session, but generally, like we are not having question
Thank
answers.
you.
Yeah.
We will now felicitate our orator. Thank you, everyone. We'll have a tea break for 20 minutes now. And there is an announcement. We have got this Titan watch. It's lost. Can please, is there anyone who wants to claim this watch? This is a Titan watch with a brown band. Thank you.